This comprehensive protocol instructs how to generate closed genomes of diverse urinary bacteria and enables readers with no coding experience to successfully implement terminal-based genome assembly and analysis pipelines.
Complete genome assemblies enable the discovery of genes contributing to colonization and virulence among commensal and uropathogenic bacteria. Closed genomes provide insight into genome architecture and mobile genetic elements. The hybrid genome assembly method can be applied to any culturable bacteria isolated from other anatomical sites or environmental niches to provide similar insights into genome content and architecture.
Follow the protocol and manufacturer guide to prepare a double-stranded DNA pool and proceed to clean up by adding 2.5 times the volume of paramagnetic beads to the DNA pool and then gently flicking the tube to mix the contents. Briefly spin down the sample at 2000 times G and place the sample on a rotator for 5 minutes at room temperature. Then pellet the beads on the magnet. Next, wash the pellet twice with 250 microliters of freshly prepared 70% ethanol in nuclease-free water without disturbing the pellet. Leave the tube on the magnet during washes. Aspirate ethanol after each wash. Then briefly spin down the sample.